You did not. Especially yesterday, you did not.
And no, actually I don't remember I was angry with you at all. But related to having a discipline and actually being ordered, I do not actually joking and kidding with anyone. It's different from being angry, you know. If I'm serious related to your classes, how you're learning, you're studying, it's different from being angry. I'm so quiet, and actually I love all of you, and I actually wanted to help all of you to study better, but it's different from actually, yeah. Today we have a 2 session related to NPN metabolism. With Dr. Said Ibrahim, actually during that session you learned amino acid metabolism. We have different compounds which they contain nitrogen. Some of them they are amino acid and protein. You learn amino acid and protein. But in addition to amino acid and protein, we have another compound which we call them non-protein nitrogenous compound. We have more than 30 different compounds which they belong to this group, which is known as an NPN. Non-protein nitrogenous compound. So first of all, I'm going to talk about the heme. Can you remember what was heme? Hemoglobin actually. Yeah. In hemoglobin, we had actually a structure which inside actually can bind to the iron, but has a 4 ring. They contain nitrogen. And when we break down heme, we produce what? Bilirubin. So today first I'm talking about how we synthesize heme and how we break down it to the bilirubin and excrete it in the urine and also in the feces. So is the first part, and in the second part we had some actually nitrogenous compound which is known as a nucleotide. We had 2 groups of nucleotides, purine and pyrimidine. Remember them or?
Yes, sir.
How we can synthesize these 2 groups, and then when we catalyze them, what is produced for us? In the next session, I'm going to talk about this. But one of your friend, Ala Karim actually, she proposed in the previous time, I wanted to have a presentation about nucleotide metabolism. I said, okay, it's not actually good. She has a classes with immunology actually courses right now. For the next session, she will come and maybe 15 minutes, 10 minutes, she will speak about this part and then I will continue. So first of all, let's have a brief review on the first session, what we should know. For synthesis of heme, we need 2 intermediate, which these 2 intermediate condense together and then they lead to producing heme. We have one enzyme which is so important and known as a key regulated enzyme, especially in hepatic. And you should know each step actually is located in which part of our cells, mitochondria, cytoplasm, or other places. Related to degradation, related to synthesis, we have some disease which is known as a porphyria. Maybe 8 kind of disease we have which known as a porphyria. During the synthesis, each enzyme actually have a dysfunction lead to the one kind of porphyria. And the second part, we are going to talk about degradation of heme by heme oxygenase and glucosyl transferase. There are 2 enzymes which are so important in heme degradation. and we have a so famous disease related to the bilirubin, which is known as a jaundice, or in Arabic, yarakon. You don't say yarakon. Yarakon. Okay. Define jaundice. What is that? Actually, many type of jaundice we have, and we should know two terms: direct, indirect bilirubin. What they are, why we call direct and indirect. So first of all, again, I introduce a case. You can look at the lab results, and then in the actually end of the class, you should detect what kind of disease this patient has. These laboratory results, serum bilirubin is 9 milligram, serum bilirubin. Conjugated bilirubin 7.5, unconjugated 1.5. And also, when we are going to evaluate a liver function, we need evaluation of some enzyme, like OT or oxaloacetate or AST, is ALT, and urine bile salt, bile pigment, urobilinogen, and feces stercobilinogen. You don't need to actually right now understand all of them, but just look at these three, and then at the end of the classes, you can give me the answer. So they are non-protein nitrogenous compound. Actually, they are known as a waste or end product of metabolisms. In amino acid and protein, they produce urea, which you learned in metabolism of amino acid, and creatine and creatinine, which I'm talking about them very briefly today. Related to nucleic acid, we have purine and pyrimidine. Purine, they convert to the uric acid, and pyrimidine convert to the beta-alanine and beta-amino isobutyric acid. And heme protein, they convert to the bilirubin. So, they are the concentration of different NPN compounds. Hello, Abdullah Hamad. Assalamu alaikum. Urea 45% and ammonia 0.2%. They are different components. So first of all, when and where we need heme synthesis. Heme is located in which kind of protein? For what we need heme synthesis?
For carrying oxygen. Carrying the oxygen. Carrying the oxygen.
Carrying the oxygen. For synthesis of what we need actually hemoglobin. Yeah, for carrying of oxygen. By which kind of cell?
RBC.
RBC. So RBC are made in which part of our body? Bone marrow. So we can understand in bone marrow we need heme synthesis because we need hemoglobin. One of the main sites of heme synthesis is bone marrow. But in addition to hemoglobin, we have another protein like cytochrome, which yesterday I actually teach you, or actually some specific enzyme, they contain heme. What kind of our body is important for protein synthesis? In our body, which organ is responsible for protein synthesis? Liver. So, liver and bone marrow are two main sites for heme synthesis in our body. Heme synthesized actually from porphyrin and iron. And when degraded, it's degraded to bile pigment and iron again. We have a heme protein, and these are known as a porphyria. And as I mentioned, jaundice is due to the overproduction of bilirubin. This kind of protein, hemoglobin, myoglobin, cytochrome P450, catalase, nitric oxide synthase, and peroxidase are some example of the enzyme and protein which they contain heme. And for example, 6 to 7 grams of hemoglobin each day synthesized in our body. 6 to 7 grams of hemoglobin each day is synthesized in our body. Porphyrin, actually, they are compound or complex of metal ion with porphyrin. What is porphyrin? Is a pyrrole. Pyrrole contains one nitrogen and hazard is okay. If we join 4 of these components together, and then we join them by this way, is porphyrin. And actually they are pyrrole and 4 of them known as a tetrapyrrole. They have a 4 nitrogen group. Some actually protein also chlorophyll in the plant is somewhat similar to this compound. But instead of iron, they contain magnesium. Related to vitamin, one of the vitamin structure was similar to this compound. Which one?
This.
B12.
B12. B12 contain cobalt instead of iron. And actually the ring known as a chlorine, but somehow is similar to porphyrin. So here in the middle of the actually this ring, iron can be located. And iron can have a 6 bond. 4 with this nitrogen and 2 are remain with oxygen, one of them with oxygen, and the second with one of the histidine of the protein structure. So for this reason, it can actually transfer oxygen.
So because of the it's can contain like 6 atoms. So
It can bond with 6 different atoms.
Yeah.
Each protein can transfer one oxygen, one molecule.
No.
Each hemoglobin,
Yes.
can react, can bind with one
One
oxygen.
oxygen.
And in hemoglobin, because consists of 4 heme group, can transport 4 oxygen.
You mean 2 atoms? That means
Let me to continue, Mohammed Hassan. Heme protein, here you can see some example of heme protein. Myoglobin and hemoglobin, they transfer and storage oxygen. Cytochrome C, cytochrome B5 and other cytochrome you saw, they are important in electron transport chain. And also we have a NOS, nitrogen oxysynthase, and P450, which they actually oxygenation reaction they participate. And also we have some protein which they are sensor protein, like hem sensor and base sensor. So all of them, they contain heme. Here you can see some example and their function. Hemoglobin function is transport oxygen. Myoglobin is important in the storage of oxygen. Cytochrome C in electron transport chain. Cytochrome P450, hydroxylation of xenobiotics, catalase, degradation of hydrogen peroxide, and tryptophan pyrrolase, oxidation of tryptophan. There are some example of the heme protein. So just hemoglobin is not important. All of them are important, but hemoglobin synthesized in a huge amount. Daily 6 to 7 grams of hemoglobin just synthesized. So we know we have a 2 main sites. One of them is liver, and the second is bone marrow. Liver for heme protein like CYP, as this bone marrow or erythrocyte producing cell in bone marrow. They produce heme in bone marrow, and near to more than 80% of hemoglobin or heme synthesis occurs in the bone marrow. So the main site also is bone marrow. Here is a structure of porphyrin. You can see we have iron and 4 pyrrole. This is pyrrole, 4 of them together, they can create a pyrrole. But how we can create this pyrrole and tetrapyrrole? First of all, we need 2 compounds. They are what? Glycine and succinate CoA. Okay. These 2 molecules together produce another compound which is known as an ALA, aminolevulinic acid, or ALA in brief. The enzyme, because synthesized ALA, known as an ALA synthase or ALA synthetase, because actually combine these 2 molecules together, remove coenzyme and CO2, and ALA is synthesized for us. We have 2 kind of ALAs. ALAs 1, ALAs 2. It was the first reaction. This reaction occurs in the mitochondria. Okay. Then here you can see ALAS1 actually is a mitochondria, 2 also is a mitochondria. Can be inhibited by heme. Heme is the final product. When we produce heme enough, we don't need more heme. So heme itself come and inhibit heme synthesis. Is related to ALAS1, which located in the liver. In the liver, we can regulate heme synthesis. But ALAS2, which is located in the bone marrow, cannot be inhibited by him. Just iron is important. If the sources of iron be enough, we can synthesize heme as much as we want. The next reaction, we have a glycine and succinate combined together. Then we send out of the mitochondria to the cytoplasm. We combine 2 molecules of ALA, together, and we remove a water hydrate. So when we remove water, we call it dehydratase. Because the substrate is ALA dehydratase. And what is produced for us? This molecule, which is known as a PBG. So 2 molecules of ALA together by ALA dehydratase.
Which type of ALA?
What?
Which type of
Delta aminolevulin. What do you mean which type?
ALA? Is it 2 types? 1 and 2?
No, ALA is the enzyme. We have a 2 type, not the product. Product is similar. We have a 2 kind of enzyme. ALA dehydratase remove 2 molecules of water and then produce PBG, or other name is porphobilinogen. Porphobilinogen. Okay, PBG. PBG, if you look at here, is similar to the pyrrole, but also has some branch. These branch are important to produce different kind of porphyrin. And based on this branch, we can categorize porphyrin. Is the precursor of pyrrole, the first precursor. This actual reaction occurs in the cytosol. So again, you can see aminolevulinic acid, 2 molecules of it can be actually converted to the porphyrin. And we have a 2 molecule of water, and the enzyme name is the hydratase. Who is responsible for inhibiting of this enzyme? Lead. Lead. What is lead in Arabic actually? Rasas. Rasas. Okay, it's so dangerous, especially for children and somehow for girls. Can you give me why, the reason of why is for girls and child?
Is it because they have less hemoglobin than grown males?
What?
They have less. Is it because they have less hemoglobin?
No.
Does
Let's-
it deplete?
Just thinking about it, I will talk about it. Thinking, just 5 minutes thinking, then I talk about it. So we have a PBG. Here we have a propionate. Here we have an acetate and 1 methyl and amino group here. So the next reaction, we remove this amine group by an enzyme which name is hydroxymethyl synthase. But this reaction is not so important for us. 4 molecules of PBG together combine and produce this type of uroporphyrinogen. Okay, 4 molecules of PBG produce uroporphyrinogen. How many molecules of glycine we need for porphyrin synthesis?
One.
How many molecules of
Glycine.
we need?
One.
1. Agree?
How is it getting them?
So 4 PVGs make
Make porphyrin, yeah.
just one.
Just 1 glycine we need?
No,
Four.
we need 4.
4 glycine.
For nitrogen.
Yeah, it's because of
8. Why 8? Because here, for the first time, we need 1 glycine and 1 succinyl to produce 1 ALA. And again, we need 2 molecules of ALA, so we need 2 molecules of glycine. Then we need a 4 molecules of PVG, so if you multiply 2 to 4, it gives us 8. Homie, 8, not 1. So, uroporphyrinogen is somehow porphyrin, but it needs to actually do some change. Here we have acetate propionate on the structure. Next reaction, we have a decarboxylase. Uroporphyrinogen decarboxylase. Remove a 1 methyl group from the acetate and convert them to the methyl group. Uroporphyrinogen decarboxylase. 4 CO2 from this acetate group. Remove and cupropurfirinogen produce for us. So this cupropurfirinogen again sent to the mitochondria. So till now, our reaction occurred in the cytosol. But cupropurfirinogen again sent to the mitochondria here. And then we have a 2 level of oxidation by these 2 molecules. And finally, we have a protoporphyrin. In the last reaction, ferrochelatase add iron and produce heme. This enzyme also can be inhibited by lead.
So far, 2 inhibitors, but lead.
Yes, islate dehydratase and ferrochelatase, both. So related to side chain, you saw we have acetate propionate, methyl propionate, and methyl propionate and vinyl. They are different side chain. And related to distribution, I told you. So Which enzyme catalyzes the condensation of two molecules of
A.
ALA and form purphobilinogen?
A.
The first one.
A. Convert two molecules of ALA to form purphobilinogen. Why you don't understand the question?
B.
What
B. ALA dehydratase or PBG synthase. The next question. Which enzyme introduced iron into the porphyrin type 9?
C.
C. Ferrochelatase. So, there are some actually example of questions. Here, you can look at the whole reaction together. This is mitochondria and this is cytosol. The first reaction, glycine and succinyl-CoA together by ALA synthase produce ALA or amino levulinic acid. Send it to the mitochondria. Two molecules of ALA together produce PBG.
Using
Four molecules of PBG produce uroporphyrinogen. Then uro convert to the cupro. Cupro again sent to the mitochondria. Proto, porphyrin, receive iron and actually produce heme.
What are those letters on the side? ADP, AIP, CEP.
What they are? It's so actually good question. They are the name of disease. Each of these enzymes actually has a dysfunction lead to these kind of disease. The first one known as a cytoplastic anemia, X-linked protoporphyria. X-linked protoporphyria. Okay? And for example, here we have a cutaneous erythropoietic porphyria. I will talk about the name. But you don't need to memorize all kind of diseases. Those are important for me are here. The first one, ALA synthase. If we have a dysfunction in ALA synthase, actually, it's known as a cytoplastic anemia, the first. Because we learned we need heme for RBC, for producing blood, actually, and if we have a dysfunction lead to the anemia, is the first one. And the second is related to liver. If we have a dysfunction in heme synthesis, we have problem in abdominal, okay, in abdominal part. So we have some porphyria related to anemia, some porphyria related to GI tract problem. This is related to anemia. And this enzyme, ALA dehydratase and ferrochelatase, both of them are inhibited by lead. And actually, uroporphyrinogen decarboxylase also is important because it's the most common type of porphyria and actually lead to the porphyria cutanea target. But there is some point to actually memorize all kind of molecule in the heme synthesis. They are the first letter of the whole structure. Get some additional point having understood the correct pathway for heme synthesis. If you actually remember that, also I think it's more difficult than memorizing the molecule. And it's so good actually sign letter for understanding the whole molecule here. Related to ALA synthase, I told you we have a two kind of ALA. ALA S1, ALA S2. ALA S1 is found in all tissue, okay, especially in liver. And ALA S2 is just in bone marrow. So ALA S2 in bone marrow, ALA S1 in all tissue. And ALA S2 lead to the X-linked sideroblastic anemia. ALA S1 is a rate limiting enzyme for heme biosynthesis. ALA S1 is a rate limiting enzyme, is a key enzyme in the heme biosynthesis, just in liver, not in bone marrow. And ALA S1 also has a short half-life. But related to the actually the ALA S1, because it's important in the liver. And cytochrome CYP. Did you hear this molecule before? Cytochrome P450.
Yes, you mentioned it.
You know what is the function of this kind of cytochrome?
No.
The metabolism of xenobiotics, drug, poison, all of them are actually metabolized by these enzymes. We have more than 250 molecules of cytochrome P450 for metabolism of all kinds of drug, poison. So when we actually use some drug, because we need this molecule for synthesis of heme is increased because this molecule contains heme. So many drugs whose metabolism require cytochrome P45, they increase the biosynthesis of heme. Okay? And actually the depression of intracellular heme produce and induce heme synthesis. But ALAS2 is not feedback regulated by heme. You got it?
Just that one.
The one related to 2 cannot be inhibited by her because we need heme synthesis all the day, all the time, all the condition. Salam. Salam. Salam alaikum. Salam alaikum. Why you don't pay attention to the time of the class? What are you doing on the floor? Actually, can be regulated just by iron, but is not regulated by the heme because all the time we need ALAS2 for heme synthesis, for hemoglobin, for producing RBC. They will produce a large amount of the RBC. So we have a 2 term related to heme production. Some molecule in the suffix they have a in, like furfubilin, urobilin. Some of them they have a ogin, like furfubilinogene, orobilinogene, cuprofurfurinogen, and so on. Those they have a ogin, they are colorless. Okay, and those they have a in suffix, they are color. And this is the actually method to detect bilirubin and other kind of actually metabolite by the measurement of the wavelength in 400.
Sir, does this suffix gen also mean synthesis or not?
No, it's not. That is genesis. Genesis is a synthesis. Glycogenesis, gluconeogenesis is different from the ogin. Is ogin.
What about ribsinogen?
They are zymogen is different from ogin. They are zymogen. Okay, it's somehow also they are different. Zymogen like chymotrypsinogen, trypsinogen, and other enzymes. They are just for actually heme product like cuprofurfurinogen, furfubilinogen, orobilinogene, and so on. That's these 3.
What about myoglobin?
What?
Myoglobin.
Myoglobin. Actually, myoglobin contain one heme, and like hemoglobin, actually is synthesized by ALAS4. ALAS2, sorry. But the function completely is different from hemoglobin. As you learned in previous semester, myoglobin just is important in the muscle cell. In the muscle, close to the mitochondria. And just act as a storage of oxygen. We don't talk about myoglobin too much, but heme can be used for hemoglobin or myoglobin or other protein. So, related to disorder of heme biosynthesis, we have some genetic or acquired disease. Lead poisoning lead to inactivate of ferrochelatase and ALA dehydratase, as you learn, and we have a different kind of porphyria. So all kind of porphyria together, it means we have a problem in heme synthesis. Here, if we have a dysfunction of ALA synthase, because it needs B6 as a coenzyme, if we have dysfunction in ALA synthase or dysfunction or actually insufficiency in B6, they lead to the sideroblastic anemia. Lead poisoning inhibit ALA dehydratase and also ferrochelatase. Acute intermittent porphyria is due to the dysfunction of PBG deaminase and porphyria cutanea tarda PCT, AAP, XLP, like that the names. They are actually cutanea porphyria, which is actually somehow has a complication in it. But my question was that why in the child and girls heme synthesis is important and also lead to the lead poisoning, lead to the heme dysfunction. What do you think?
Because hormones like because the
No,
girls and
you
child.
actually thinking so complex.
Is it at a higher rate in both children and girls, and that's why the lead combines faster in the lead poisoning
No.
treatments?
Actually,
Is it
I,
because it's
actually, I was know that it's dangerous for the children because it's infection on their developing brains and the nervous system. But what's the difference between the infection of the lead on the male and female?
It's not an infection, you know, it's related to
It's
the...
related to what you
is
see.
it because women and children have less orgasm?
No.
It's related to
Somehow, yeah, but not the reason. You know, child, actually, they have actually tendency to try everything. If you give them any toy, they try to taste it. And most of the toy, they have lid, okay? Especially those, what you say, we call it, yeah, slime. Yeah, slime, exactly. They contain lid. And when they actually try to taste them, it actually lead to the lid going to their body and lead to the heme dysfunction.
And
Sure.
women
And what
also
about the women?
They also taste everything we give them.
What they?
Is it because women also taste everything?
No, they don't taste it.
Because for
Because the
women
makeup-
it's stored in rooms.
What?
It's stored in rooms because of that.
What?
Because of work.
No.
Cosmetics.
Cosmetic. What kind of cosmetic?
Lipstick.
Makeup. Which one?
Lipstick.
Lipstick.
Lipsticks and
Lipstick also contain lid.
concealers.
Powder
Is
as
the
well.
reason. Lipstick.
Powder as well.
What?
Powder.
Powder.
Yes, they used in England. They used to
Powder,
have
but cannot actually go inside of the body. But lipstick
برضه
actually
عشان يخلون
is more.
شو اسمه
And
powder على
because of
وجههم
the child,
فيتسوون.
also their bodies absorb the lead more than the adults.
Yes, somehow is a lid, but the lid goes to their body more in comparison to others. So, sideroblastic anemia is a kind of anemia which known as a congenital due to the ALA synthase deficiency. What's happened? It can be due to the B6 deficiency using ethanol or lead poisoning. Also, we have a drug which is known as isoniazid. Isoniazid
B6 deficiency.
lead to the B6 deficiency. Why we call it sideroblastic anemia? Did you hear this term, sideroblastic anemia?
It has something to do with development, making
No.
cells on the side.
For heme synthesis, we need iron. Okay? If heme synthesis reaction, for example, ALA synthesis, doesn't work, we cannot synthesize heme. So iron is accumulated. Where?
Mitochondria.
In the mitochondria. And when iron accumulated in the mitochondria created a ring, a sideroblastic ring, which actually somehow is blue. It's known as a sideroblastic anemia. The iron amount is enough, also is too much, and accumulated and lead to the actually this kind of complication. Why? Due to the inability of iron to form heme, trapped in the mitochondria and accumulated. What are the reasons? Actually, the lab result is like that. Ferritin is increased, TIBC decreased, iron also is increased. The treatment is pyridoxine. What was pyridoxine?
B5.
Vitamin
B6.
B6. And lead poisoning actually also has its salt, and here you can see lead to the accumulation of lead, especially in the lip.
The sideroblastic, yeah. Which type of the AL synthesis? 1 or 2?
Which one?
You say ALA synthesis. Yeah, which type? 1 or 2?
Which
You
one
say,
is important for us?
2.
No, for anemia.
For anemia? 1.
Yes. One?
No?
Both of them important.
No, for anemia, for
Two.
RBC.
One.
Two. Two. For blood is 2.
Does it work all the time in the bone marrow?
What?
Because the bone marrow work all the time, not inhibited
You
by
know, ALAS, I told you we have a 2. The first one is located in liver and all
All
tissues.
tissues.
2 is in
In
bone
blood.
marrow.
Only bone marrow.
And in bone marrow, we have more than 85% of heme synthesis. The first reason. Then the second, when we talk about anemia means we cannot produce
Enough
cell blood enough.
Yes.
Cell blood means RBC. RBC need heme. Heme is synthesized by ILA synthase. So, actually also lead can be accumulated in the joint, and in X-ray, in the graphy, it's completely clear they actually have this kind of discs. The next, this is known as acute intermittent porphyria, which due to the dysfunction of PDG deaminase, and some drug like phenytoin, barbiturate, sulfonamide, and actually they induce this disease. Porphyria cutaneous tarda. Cutaneous means skin, So we have a problem in their skin. Actually, we have blistering, hyperpigmentation, and most of the porphyria. Asan, I'm talking about what? What kind of porphyria? What kind of problem we have?
Thinking about it.
Hmm?
The causes, association, and treatment. It's mostly deficiency.
Deficiency in what? Euro?
I can't say it.
You can say. Actually, most of the porphyria, because if you look at that, the structure is a tetraphyllid, and it's so difficult to excrete it from our body. And when actually light, especially in the skin, receive to this molecule, lead to the destruction of the molecule, and lead to the creating a pain. So they are photosensitive. Porphyria, they are so photosensitive. They prefer to live in darkness because light lead to the degradation of this molecule and creation a lot of pain. So most of the porphyria, they have a photosensitive. It's kind of actually a patient with porphyria cutaneous tarda. You can see blistering and hyperpigmentation in their skin, and also their urine change, the color change to the red somehow. They are the sign and symptom. They have abdominal pain, anxiety, depression, paralysis, motor and sensory, actually autonomic neuropathy, weakness, and excretion ALA. When they use some drug like barbiturate, the episode also are increased. Here you can see all kind of 8 diseases related to porphyria. Among them, the first one, ALA synthase, which lead to the anemia, is important for us. And actually, uroporphyrinogen, the carboxylase, which lead to cutaneous tarda, and lead poisoning lead to the ALA dehydratase and ferrochelatase. And if you look at that from here, we have photosensitivity in all kind of disease, and also abdominal pain. Abdominal pain and photosensitivity, there are 2 actually complications of porphyria. Again, you can see all enzymes and their complication and the name of enzyme. For example, porphyria cutaneous tarda, PCT, congenital erythropoietic porphyria, CEP, acute intermittent porphyria, AIP, and all of them are here. So, porphyria cutaneous tarda is associated with deficiency of which one?
Urination.
Which one? A?
Decarboxylase.
No.
Then C.
The
C. Lead poisoning is associated with severe deficiency of
LA
ALA synthase.
yes.
No, I think B.
Dehydratase.
Dehydratase. B.
So 5-minute rest. 5-minute break.
Speed.
Is the answer B?
Listen what I'm saying. No, I don't say the result because you don't listen very well. 5-minute break. Completely clear.